In this session, we are going to learn about the C4 pathway or the C4 cycle. So, the C4 pathway or the C4 cycle is a special type of cycle which is found in special type of plants called the C4 plants, in which the carbon dioxide will be fixed in a unique way. How it will be fixed, we will see just in a bit with the help of a diagram. First, let us understand what are C4 plants. So the C4 cycle occurs in the C4 plants, and this C4 cycle is also known by the name Hatch and Slack pathway. Hatch and Slack pathway. Alright, and the C4 cycle will be seen in the C4 plants. Now, what are C4 plants? The C4 plants are different from the C3 plants. C4 plants have a unique feature inside them, specifically inside their leaves, which is called Kranz anatomy. Kranz anatomy is present in the leaves of C4 plants. Now, what is this Kranz anatomy? To understand that, we will look into the internal structure or anatomy of a leaf of a C4 plants. Alright, so Kranz means wreath. If we look at the word Kranz, it means wreath, and it is associated with the arrangement of cells. Arrangement of cells specifically around the vascular bundle. So let us look at this Kranz anatomy. So if we talk about the anatomy of a leaf of C4 plant, then if this is the vascular bundle in the middle, then around this vascular bundle there will be several layers of bundle sheath cells, which will be very thick and impervious to gases, and there will be no intercellular spaces between these cells, so they will be very tightly packed. Alright, so this is the bundle sheath. Bundle sheath is a covering of a layer or several layers of cells surrounding the vascular bundles. Alright, so in Kranz anatomy, we will see that there will be several layers of bundle sheath whose cells are very thick, and they will be impervious to gaseous exchange. They will not allow any gases to pass through them, and there will be no intercellular spaces between the cells. Now, above the layer of this bundle sheath, there will be another layer of mesophyll cells arranged like this. So these are the mesophyll cells surrounding the bundle sheath of vascular bundles. Mesophyll cells. Now the number of chloroplasts in the bundle sheath will be larger as compared to the chloroplasts present in these mesophyll cells. So this constitutes the Kranz anatomy of the C4 plants, which basically refers to the special arrangement of bundle sheath and mesophyll cells around the vascular bundles of the leaves of the C4 plants. Alright, and this Kranz anatomy is seen in the C4 plants, and the C4 cycle or the Hatch and Slack pathway is specific to this Kranz anatomy. So where and how will this pathway occur, let us try to understand. Now before that, let us talk briefly about the C4 plants. Now these C4 plants are special plants that grow in a dry tropical climate. That means they are adapted to tolerate high temperatures. They can respond to high light intensities. High intensities of light. They lack photorespiration. What is photorespiration? We will study in the next lecture. For now, you just have to remember that photorespiration does not occur in C4 plants, and they have higher mass productivity. Or we can say that they are more efficient in photosynthesis as compared to C3 plants. Alright, so this was about the C4 plants. They typically grow in the dry tropical climate and are adapted to tolerate high temperature. They lack photorespiration and are more efficient in photosynthesis, and the leaves of the C4 plants bear this Kranz anatomy, where we will see this special arrangement of bundle sheath and mesophyll cells around the vascular bundles. Alright, now let us understand the C4 cycle. So in the C4 cycle, the first acceptor, the primary acceptor of CO2 is phosphoenol pyruvate. Phosphoenol pyruvate or PEP. This is a 3-carbon compound. Alright, so this will accept carbon dioxide from the atmosphere, and it forms the first stable product, which is oxaloacetic acid. Now oxaloacetic acid is a 4-carbon compound. Alright, so the first stable product here is the oxaloacetic acid, which is the 4-carbon compound. Now this oxaloacetic acid is converted into malic acid or aspartic acid. It's converted into malic acid or aspartic acid, and then it is transported to the bundle sheath cells. Alright, so the reaction that we are studying here till the formation of malate or aspartate, this is occurring in the mesophyll cell. Alright, so this is occurring inside the mesophyll cell. So here, in these mesophyll cells which are surrounding the bundle sheath, this reaction is occurring in the mesophyll cells up to the formation of malate or aspartate. Now once the malate or aspartate is formed, they are transported to the bundle sheath. So now they will be transported to the bundle sheath, and here they will lose carbon dioxide by a process of decarboxylation. And they will form a 3-carbon compound. Alright, and this 3-carbon compound from the bundle sheath, this is happening in the bundle sheath. Okay, this is the bundle sheath. Decarboxylation occurs in the bundle sheath, carbon dioxide will be released and the 3 carbon compound will form. This 3 carbon compound will again enter the mesophyll cell. It will be transported to the mesophyll cell where it regenerates the phosphoenol pyruvate, which is the primary acceptor of carbon dioxide. So here the regeneration takes place. This is the regeneration step of PEP. Now here I forgot to mention the enzyme. The phosphoenol pyruvate, it accepts the carbon dioxide, that is the carboxylation takes place in the presence of an enzyme called pepcase or pep carboxylase. Alright, so the phosphoenol pyruvate carboxylation occurs, oxaloacetic acid is formed, which is a 4 carbon compound converted into malate or aspartate in the mesophyll, then transported to bundle sheath where the decarboxylation of malate or aspartate takes place and a 3 carbon compound is formed. This 3 carbon compound is transported back to the mesophyll where it regenerates the phosphoenol pyruvate. So this constitutes the C4 cycle. Alright, so the C4 cycle occurs in the mesophyll as well as in the bundle sheath. So this was the C4 cycle. Now the sugar is not yet formed, the carbohydrate is not yet formed. So how it will form? In this step in the bundle sheath, when the decarboxylation took place, the carbon dioxide was released. Now this carbon dioxide will enter the C3 cycle inside the bundle sheath. Alright, so this CO2 will enter the the C3 cycle inside the bundle sheath, the Kelvin cycle, and all the steps that we studied in the Kelvin cycle will occur here in the bundle sheath. The bundle sheath has the enzyme Rubisco. We know that Rubisco is involved in C3 cycle, right? So this enzyme is present in the bundle sheath. So once the carbon dioxide enters the bundle sheath, then C3 cycle takes place and there will be formation of carbohydrate or starch inside the leaves of the C4 plant. Alright, so one thing to notice here is that although C4 cycle is going on, the main biosynthetic pathway here is the C3 cycle only, because with the help of C3 cycle only the carbohydrate is produced in the C4 plants. Alright, that's why when we were studying about the C3 cycle, we studied one point that C3 cycle occurs in all the photosynthetic plants, whether they are C3 plants or C4 plants. Even in C4 plants, C3 cycle will occur along with the C4 cycle. Alright, and the main reason why all this happens in mesophyll and bundle sheath separately is to avoid photorespiration. The C4 plants, they try to avoid photorespiration, and that is why this special type of adaptation is found in them, which is Kranz anatomy, and the C3 and C4 cycle both will occur. Alright. So it is important to remember here that in C4 plants, the mesophyll cells lack Rubisco enzyme, and Rubisco is found in the bundle sheath. Therefore, C3 cycle occurs in the bundle sheath, and all this special arrangement is to avoid photorespiration. Alright, now the example of C4 plants are, there are three examples of C4 plants: maize, sorghum, and sugarcane. Alright, and C3 plants will be those plants in which the C3 cycle occurs. Alright, and C4 plants are those plants in which the C3 and C4 cycle both occurs. to avoid photorespiration. And these are the examples of C4 plants. In case of C4 plants, rubisco is present in the bundle sheath and C3 cycle occurs inside the bundle sheath. Whereas in case of C3 plants, rubisco is present in the mesophyll cells. So all the mesophyll cells are involved in carrying out the C3 cycle. So I hope C4 cycle or the Hatch and Slack cycle, as well as Kranz anatomy and C4 plants, all these topics are clear to you. That's all for this session. Thank you so much.